So for those of you guys that don't really know me, I'm Michael Zhang. I'm doing a talk on From DNA's Discovery, which is pretty much just a foray into how I got involved with research. And I'm going to be throwing in a couple things about the whole discovery process that I've kind of realized over time. So yeah. So I want to start off with this molecule, DNA. It might be a little strange to think of when I say that I feel like I'm very good friends with this molecule. Yeah, it's a little weird. I could tell you a lot though about it, like how it has all these purines and proteins and 4 base pairs and all that stuff, which isn't really that important to our talk. But it's something that I think is really important because DNA is kind of what got me interested in biology and research in the first place. So yeah. Before we go into that, I want to talk to you guys about one discovery tip that I guess first learned about, which is the challenge assumptions. Now, hands up, who here has ever really, I guess, learned something or thought of something, and it was just like kind of like forced onto you, like, I don't know, people used to say that the earth was flat or something like that, and you just believed it. Just like, I don't know, it happens in school a lot. Lots of people are just like, oh, just do this, we're gonna maybe learn about it later. But, what I want you guys to do is to never really look at that statement and just accept it. I want you guys to challenge assumptions. Like even this statement. Don't believe me just because I'm up here talking to you guys about it and saying that it's a good thing. Think about it, and after you think about it, if you accept it after that, that's fine. But before that, don't do it. But moving on. The experiences that I keep talking to you about are, of course, my forays into research. And as a freshman, I got the really cool opportunity to visit a lab at UPenn, the medical school actually, and got my hands into like this really cool biological research. And being the ambitious little kid that I wasn't really satisfied with just being here and just being babysat by all these like really old scientist guys. I wanted to actually do something about that, and I think the first thing that I said was like, I'm going to spend this summer and I'm going to cure cancer. Really, really ambitious, but also probably disillusioned and stuff like that. But the important thing about this was that people told me, of course, that I probably couldn't do that because I was too young, too inexperienced, didn't really know how to do anything in lab. But I kept myself motivated by challenging that assumption. Now, okay, we already talked about this. Who cares ever believe something just because, again, don't do it. So yeah, so people are like saying like, oh, what are you doing here? I'm just like this little kid here. They're like, you can't really do anything. But using that belief of challenging assumptions, I came up with this whole like project proposal and all that. Weapons of precise destruction. Engineering novel SNIP-like bispecific fusion proteins to combat cancer-induced osteoporosis and tumor proliferation in multiple myeloma. This is actually a really terrible title, but what I learned from this experience was that it wasn't about just like me being inexperienced at the first place. If I put in the effort and I could actually kind of be creative about things, I could turn out something that was actually pretty legit. And that goes into discovery tip number 2, which is you're never too underqualified or young to do something. If you've got an idea or a passion, you shouldn't be afraid to pursue it to a further extent than you actually are right now just because you feel underqualified. And if you actually are underqualified, do everything in your part to be able to get to that stage where you are confident about yourself. So what actually, you don't see that slide yet. So what actually happened? Turns out that people were right when they said that I couldn't do it because, yeah, bad things happen in the lab when you don't know what you're doing. I had a really bad incident over the summer where I constantly break Bunsen burners, and luckily that hasn't ever happened in any of the chem labs I've been at for 2 years, but that summer was really memorable because I ended up getting a lot of burns on my hands. But what's important to know is that I continue to maintain this idea that you should challenge assumptions, because through all that burnout and all that stuff, while other people could have been like really frustrated or depressed and whatever, I realized that this was just part of the process. Before you can get success, you got to go through some things, I guess. And I decided to think about my stuff more and revise things, and then I ended up coming up with a new proposal to, I guess, further my research and stuff. And that ended up going onto this molecule. Now Lucas actually did a really good job introducing it. It is Cas9, part of the CRISPR-Cas9 system. And what is really cool about Cas9 is that, this is CRISPR-Cas9 by the way, is that it is able to basically go into any DNA and cut out the genes that you want, or turn on certain genes and turn off others. And this is actually really, really impressive. It doesn't sound that important unless you're like a bio teacher or whatever. But if you think about it, any type of cancer or disease that is labeled incurable is mostly because of a genetic defect in you. Like drugs can't help you if you've got something fundamentally wrong with your DNA. But anyway, if you think about it, using CRISPR, scientists and doctors could one day go into a single cell, and what they could do is target that specific DNA and pretty much cut it out. So you can cut out your cancer mutations. That's something that's really cool. It's far away, but it's definitely something that's possible. And it's also a reason why CRISPR is so much cooler than toast. But moving on. The problem with that idea, though, is that for any sort of medicine, you have to think about how you can get it into people. There are studies where they did it in mice, where they pretty much shoved all this liquid into them, and it ended up like the mice got their genes changed, but they also ended up like ballooning like 200% because their bodies were made of, or like people inserted like pretty much 100% more volume into them. And humans can't really do that because if that happens, they die. So what I tried to figure out was how we could get this CRISPR system into people so that it could be used for medicine. And I thought about it, and I got an idea. I was going to go against millions of years of evolution and re-engineer viruses to not infect people and do all this bad stuff, but to instead perform CRISPR in them. And it sounds like a really far-fetched idea, but if you look into it more, viruses are already really good at getting into cells. That's the whole reason they make us sick all the time. If we could just figure out how to remove whatever is making us sick in there and just replace it with this CRISPR-Cas9 system, maybe we could be onto something. And it turns out that when I did this research, the virus that I was actually using was a very close cousin to HIV. And so the idea here is that we think of HIV as a really, really bad thing. It causes a really, really bad disease, AIDS. But although it's far-fetched to say that I was using AIDS to cure cancer, there is a correlation between that and the idea is to think differently, but not too differently that you throw reason out the window. So yeah, be practical about your stuff. So I was like, cool, really, really cool idea. I'm going to submit this to Penn. My professors are going to love it, and I'm going to be able to do this research and become famous. Well, it turns out that first professor couldn't do it because just didn't have a lot of stuff in the lab to be able to do that. So I went to Penn Engineering's website and emailed all of these people. Turns out that not a single one of them wanted to do this. And I was like, why? Why do you not want this? Okay, this is a good thing. So I was a little frustrated at first. But yeah, they're going to school and I'm not there, but whatever. What was good about it is that I want to go back to the slide because I went back, I thought about it some more. I used people skills. People skills are really, really important. Never forget your people skills. Be able to talk to people. I used my people skills, and I was able to take that research and send it to other people beyond Penn. And I somehow ended up there, which is not MIT, but Harvard Medical School. And from there, I was able to spend 2 really great summers at this really great place called the Beast Institute. I'm wearing a shirt there, self-promotion there. But there for 2 summers and do some really cool research, meet some really cool people. The guy here is my professor. He kind of looks like Santa Claus, and that was why I guess that kind of stood off to me at first, but he is a genetics professor at Harvard Med. He really got into CRISPR research and all that DNA sequencing and all that stuff that Lucas also mentioned. And I got to see if my viruses would actually work. So this is just a figure to one of the papers that I recently worked on. Yeah, you first gotta look at the proteins, and then you gotta like put them into cells and all that stuff. And there's a lot of work to do on stuff, but it turns out that they do kind of work. So here you can see that this is like cells without anything like CRISPR or whatever, and this is cells with CRISPR. And in this case, green is good. Green is go, so yes. It was good. I ended up getting like 200% more expression, so that was cool. And just going back to this idea of from DNA to discovery, it wasn't really something about like how I was like, I don't know, super smart or whatever, because I'm not. What was the case was that I had this, I guess, idea to do something, and I wasn't afraid to challenge assumptions, think about how I could be qualified to do it, and really think differently. That got me to where I am. And I urge all of you guys, any sort of passions or whatever interests or something like that, to just consider what I've said here and think about it and kind of follow through with that stuff. Because if you do, I promise that cool things will happen. All right, thanks guys.